Good afternoon, and welcome to today's Chardan leadership call featuring Senti Biosciences, which is a company that's advancing cell and gene therapies using gene circuits, with their lead internal program being a logic-gated CAR-NK therapy for leukemia that we'll get into shortly. A special thank you to our guests. We have Dr. Tim Liu, co-founder and CEO, Jay Cross, the CFO, and Dr. Kanya Rajangam, president, head of R&D, and CMO of Senti. And I'm Geulah Livshits, a biotech analyst at Chardan. And during this call, we'll not be discussing any Chardan research. Any discussions about research should be coordinated between our participants and their respective salesperson at Chardan. And the recording of this call will be made available to clients afterwards. And our compliance team has asked me to read this statement. By participating in this call, our speakers attest that they have made Chardan aware of any potential conflicts, and they will not discuss any material, non-public, or confidential information that they're aware of that may breach their legal, regulatory, or fiduciary responsibility to any parties. All right, with that, let's get going. Thank you all for joining us today again. Before we get into the specifics of Senti 202, could you give us a brief overview of the company?
Sure. Yeah. Thank you so much for having us. We really appreciate the time and opportunity to share what Senti is doing. So at the heart of it, Senti is focused on solving one of the key problems in oncology, which is how do you achieve maximal on-target, on-tumor killing while minimizing toxicity against off-tumor tissues. And it's that issue of specificity that's quite challenging for many other modalities that are out there, including ADCs, T-cell engagers, even conventional CAR T cells that are relying on a single antigen to give you that therapeutic window or that level of specificity. Here at Senti, we are focused on using a specific type of genetic circuit that we program into a cell therapy. That gene circuit that I'm talking about is called a logic gate. And the idea is to use this technology to go after the cancer cells by recognizing cancer targets, but to also spare healthy cells from toxicity by recognizing those healthy tissues and basically modulating or reducing the activity of the product against those tissues. At the heart of it, we are a logic gate focused development company. We're about picking the right cell type for the right indication. So we have this technology working for both allogeneic CAR-NK cells as well as in CAR T cells. And we very much believe that this can help us address a wide number of indications in oncology that are not adequately addressed today by those single target products that many people are pursuing in the clinic.
Great. So maybe let's get into Senti 202 a little bit more. Can you explain the design of the asset and how it's differentiated from both other cell therapies as well as other products in the AML space?
Sure. Yeah, so Senti 202, the cell type that we're using is an allogeneic CAR-NK cell. So we source these cells from healthy adult donors. We have a whole donor selection process that's achieved by. We can expand those cells in our manufacturing facility, and we then can transduce a single virus that carries with it our genetic circuit. Then we can continue to expand the cells, cryopreserve them, and use them as a true off-the-shelf therapy for patients in need. The actual logic gate that's contained in Senti 202 has three components. The first component is an activating CAR. So this is a CAR receptor that goes after two different AML targets, CD33 as well as FLT3. And so engagement of either of those cancer antigens can trigger killing. So we call that the CD33 or FLT3 CAR receptor. Now, CD33 and FLT3 are well-known antigens and targets in AML. There are approved drugs that go after CD33 on the cell surface or FLT3 ITD mutations as an example. So we chose these cancer targets to go after because they are validated in the clinic. And by putting them together, we believe this product has the potential to target both the AML blasts and the leukemic stem cells more effectively. Now, why do we need a logic gate here? The reason for that is CD33 as well as FLT3 are known to be expressed on healthy cells, including the healthy hematopoietic stem cell. And so to avoid the bone marrow suppression that one might get if you're targeting this without a logic gate, we actually built a second gene into the product. It's called our inhibitory CAR. And what this inhibitory CAR does is it recognizes healthy hematopoietic stem cells through a target called endomucin or EMCN. This target we found through pretty extensive bioinformatics work to be highly expressed in healthy hematopoietic stem cells. and in contrast, lowly expressed in AML cells. And so it serves as a do not eat me signal for those healthy HSCs. This inhibitory CAR recognizes those endomucin proteins and basically shuts down the activity of the CAR NK product. And so that's where the logic comes in. We have a CD33 or FLT3 not endomucin circuit. The third element in this construct is an IL-15 element that allows us to further enhance the cytotoxicity and persistence of the NK cells. Unlike other cell therapies or other tumor antigen-directed therapies in AML, including ADCs, the real differentiation here is the ability to recognize those healthy cells and protect them from killing. In prior clinical studies with these other products, you have seen evidence of activity in some cases, but also significant on-target off-tumor toxicity that has really limited the ability of some of these ADC products or CAR T products to completely treat these patients or treat them more effectively. So our goal here is to really widen that therapeutic window and use our ability to shield those healthy cells from being killed to then be able to maximize that anti-tumor activity against the cancer cells.
Got it. So maybe let's talk a little bit more about the trial design and how you've moved forward with that asset. Can you walk us through the trial design, including things like the dosing regimen, and talk about how you designed that and why?
Yeah, happy to. Thank you for that question. So the trial that we are conducting with Senti 202 is a phase 1 first-in-human trial in patients with relapsed refractory heme malignancies with a focus on AML, with targets like CD33 and FLT3. Obviously, those are validated targets in the treatment of AML, so that's a big focus for us. So our trial, the patients it enrolls are adult patients who exhausted other available therapies. In fact, they have to have received prior therapies for them to come on our trial. So they must have received not only chemotherapy, but if they have one of the addressable mutations like an IDH1, 2, or a FLT3, they must have gotten that targeted therapy for them to be eligible for our trial. So it truly is a high unmet need, high risk patient population. And what we are doing in our trial is it's a phase 1. So the primary objective, of course, is safety and determining the go-forward dose, or the RP2D, recommended phase 2 dose. We do not expect going into this to have toxicities limiting our dosing because we know NKs are pretty safe, are quite safe. And so we started with exploring, we designed our trial to explore 2 dose levels where we give 1 billion CAR-positive NK cells per dose or 1.5 billion CAR-positive NK cells per dose. And then we also in our trial, looking at 2 different dosing schedules where we give 3 doses of cells 1 week apart on days 0, 7, and 14, or 5 doses of cells so that they get them twice the first 2 weeks. So days 0, 3, 7, 10, 14. Both of these, in both schedules, cells are given after lymphodepletion with fludarabine and cytarabine. So in terms of our design choices, we started with 1 billion CAR NK cells. We were able to convince the FDA, or the FDA agreed to our position, I should say, about the high safety of NK cells and the need for us to go in with the biologically active dose in order for the benefit risk to be positive, right? Considering we're giving these patients the lymphodepleting chemotherapy. So we felt good about our starting dose. And as you will see, as we go into the results, we saw responses, MRD negative CRs with our starting dose cohort. The other thing that drove sort of our choice of lymphodepleting chemotherapy is we wanted to have disease-specific lymphodepletion, so some debulking as well with the flu-Ara-C, and we had preclinical data which showed potential for synergy. What we showed is when we exposed AML cells to cytarabine, there's upregulation of the CD33 and FLT3 targets on the cells, so that when Senti 202 comes in, there is increased cytotoxicity across different effector to target ratios. So those were some of the key things driving our design element. And the final thing I'll say is we were optimistic about seeing efficacy early. So we've designed a measurement of efficacy not only by standard ELN criteria, but also for looking for measurable residual disease or MRD. In other words, that depth of response right from the beginning in our trial.
Maybe a couple of follow-ups on that. So you talked about the lymphodepletion regimen choice, and that perhaps contributing to some debulking. To what extent could the activity that you see be attributed to, the lymphodepletion regimen?
We believe very little. We do expect some debulking to come in from LD, but we've designed Senti 202 or our CAR-NK to go in and take out that last AML cell, if you will. So what we are really focused on is that deep response, right? So if you look at our clinical data, the depth of response we're seeing, all our CR/CRh patients are MRD negative up to 10 to the -4. That is pretty sensitive, right? Second, the durability that we've already presented. Our longest patient was durable for about 8+ months. Now, remember, this is, I was talking about our patient population having a high unmet need. The median survival for patients who had 1 to 2 prior lines of therapy, and then the relapse refractory AML patients is 5 months. In that setting, this durability, even with the small data set, is pretty impressive. And our response rates, we've seen 4 out of 7 patients that we presented at AACR had MRD negative CR. 5 out of 7 overall had an ORR. And all our patients except 1 had blast reduction. And in our preliminary recommended phase 2 dose, we actually had 2 out of 3 MRD negative CRs. So putting all of that together, our clinical data, the depth, the durability, the response rate, this is certainly not something that can be attributed to fludarabine and Ara-C alone, which is one component of a multi-chemo cocktail for AML patients. The other thing I want to also point out, which we presented at AACR, is in our trial, we do what we call a cytoff, where we look at, by really sensitive flow cytometry, at the cells in the bone marrow before and after treatment. One of the things we've shown in that data that we presented at AACR is if you look at these leukemic stem cells or LSCs, that's a niche population of chemorefractory stem cell-like leukemia cells, which can repopulate the marrow with leukemia and contributes to a limited durability of response. If you look at that LSC population across all of our patients, the majority of cells are in G0 phase or resting phase, which means that something like chemotherapy, which requires cells to be actively dividing because it's a DNA synthetic poison, cannot act on cells which are resting, right? The way you target resting cells is by looking at something on its cell surface, which is how Senti 202 acts. And that's the other piece of data we showed is by cytoff, patients who, when you look at LSC fraction before and after treatment, more than 10-fold reduction of LSC in all our responders. So all of that put together, we are pretty confident that what we are seeing is to a large measure from Senti 202.
Got it. And so this is an off-the-shelf NK cell asset. So can you talk a little bit about how that impacts the potential redosability, being able to do multiple cycles, and in what way that's different from an autologous cell therapy, CAR T type asset, which is definitively designed as a one-time treatment?
No, great question. So we are an allogeneic cell therapy. Our expectation going into this is to have a half-life. The way to think about allo cell therapies like ours is closer to a biologic, right? So some amount of redosing, and in fact, what our PK shows is the cells expand modestly for about 14 days. Then commensurate with the host cell immune cell recovery, our cells get cleared. So what we do in our trial is we are really focused not so much on how long is the cell present in the patient, which we don't think is the key question. The question is how durable is the response you can induce with our therapy, right? So that's what we are focused on. So the way we get there is by getting a deep response up front. And it's been shown time and time again that the deepness of the response correlates with a longer durability. Especially critical is getting rid of those LSCs, right? So Yeah, so in the way we've designed our trial is patients can get up to 4 cycles each time LD, the 3 cells or the 5 doses of cells. And that includes 3 cycles in order to achieve an MRD negative CR and 1 additional cycle for consolidation. What we saw in our trial is almost all of our patients, AML patients, they respond quickly or they don't respond, which is what we've seen as well. So our median number of cycles patients have received have been 2, with most of them seeing response with that first or second cycle. And by that, I mean MRD negative CR. We have seen in a couple of our patients response deepening from the first to the second cycle, especially in our first dose level. So that's really how we've designed our trial, right? They can get up to 4 cycles.
Got it, with lymphodepletion before each one.
Before each cycle, not each dose. Correct.
For each cycle, not within the multiple doses within a cycle.
Correct.
Understood. Yep. And so maybe just coming back to the AML patient journey, we talked about a little bit the number of treatments or the type of treatments that patients get before they get into your trial. Can you tell us a little bit more about that and the size of the population? And again, you gave a couple of the survival metrics on there. What else is there that we should know about the typical trajectory for these patients?
Yeah. So AML is a disease of the elderly. Median age is about 60, 65, something like that. So at diagnosis, essentially, in terms of their treatment, patients are depending on their fitness and their age to an extent. They are categorized into kind of older, unfit patients and younger, fit patient. The younger fit patients get as intensive a chemotherapy as they can withstand, right? So the idea is to go right up front and really clear out the disease to get to that MRD negative CR. The broader group of people who are older and unfit, the majority of AML patients who cannot tolerate as intensive a chemotherapy, they get either reduced chemo or a venetoclax-based regimen with a hypomethylating agent. But what happens with these patients is about half the patients, either they don't respond up front, okay, it's called refractory, or if they do respond, it's limited and the disease comes back, or unfortunately, they succumb to upfront therapy. Then once they relapse, the choices really are for those who have a targetable mutation to give that particular therapy. So if you have a FLT3 mutation, you can get a FLT3 inhibitor. If you have an IDH1 or 2, an IDH1/2 inhibitor. And now more recently with the KMT2A translocation, you can get a menin inhibitor. But if not, it's salvage chemotherapy. So it's really and our patients who come on, the study patients we put on at AACR, of the 9 patients, the median time from diagnosis to coming on our trial was less than a year, and they had seen 2 prior lines before coming on. So we are the 3rd line therapy for patients who were diagnosed, failed, came on within a year, right? So that's a pretty high risk patient population. And what we are seeing, one thing that has been shown in relapse refractory, the median survival is about 5 months. If you look at response rates, it's about 20% CR or so. And in terms of number of patients, we are looking at about, in the US, about 11,000 to 15,000 in the relapse refractory setting. But overall AML, I think about 21,000 patients are diagnosed with AML every year in the US.
Got it. No, that makes sense, and it highlights the unmet need in this, and kind of the pretty fast trajectory of the lines of therapy here. So again, you talked a little bit about the clinical data as we talked about the design and the rationale for the trial. Are there any other points regarding the clinical data that you shared at AACR and overall that you want to highlight?
Yeah, no, very briefly, what I'll say is, first of all, we are extremely excited about our data and our progress so far, right? We presented 9 patients overall at AACR, 7 of whom were response evaluable because they had received sufficient therapy to they've completed their treatment for us to evaluate them for best response. So out of the data we presented at AACR, in terms of patient population, I already alluded to that, 2 prior lines of therapy, vast majority had seen both fludarabine and Ara-C. Quite a few of them were refractory to one or both agents. They had all seen venetoclax in the recommended phase 2 dose. In terms of safety, extremely clean safety profile. No DLTs, no grade 3 AEIs. We didn't have a single type of SAE that happened in more than one patient. No grade 5 AEs. So very clean safety profile. What we did see were some heme events consistent with lymphodepletion or flu-Ara-C usage in these AML patients. Then in terms of dose finding and our primary objective, we've identified our preliminary recommended phase 2 dose at 1.5 billion cells per dose, given as 3 doses. That's our go-forward dose. I say preliminary because it's a 3+3 study design. So with time, we'll add more patients and confirm that. Then mainly in terms of efficacy, we saw, as I alluded to, composite CR 4 out of 7 from the evaluable patients, all MRD negative, all durable, all ongoing at the time of the data cut, longest being 8 months out. Then I think the others were 4, 5, 7, something like that in that range. And that's just because of the time they came on our trial. So super exciting. And we also presented PK looking very consistent with other cell therapies and our site-off analyses where we got insights to our gene circuitry's mechanism of action. So all in all, it was well received by the clinical and scientific community. And, we are very, very encouraged by what we are seeing so far in our trial.
Great. So maybe a couple of follow-up questions on those things. In terms of the efficacy that you talked about, can you contextualize it a little bit for us in the context of the current standard of care? And also, for example, the type of responses and activity that we see with some of the targeted approaches, other things that are in development, that kind of thing.
Yeah, happy to. So in general, relapsed refractory AML patients, CRs in literature are between about 15 to 25. So about 20% true CR rate. And then about 20 to 33% when you also include CR with partial hematologic recovery or CRH. The difference between the two is in both of them, you have blast reduction. In CR, you have counts, specifically neutrophils and platelets, recovering, quote unquote, fully up to a certain threshold. And in CRH, it recovers partially to a threshold lower than that. Sometimes in relapsed refractory AML patients, there is cumulative bone marrow toxicity from prior therapy. So there's that residual toxicity as well. And sometimes the therapy itself is also toxic because that's one of the things, as Tim mentioned with AML, the AML blasts and the healthy hematopoietic stem cells, they share a lot of their targets, so to speak. So we have our unique NOT gate designed to protect the healthy HSCs, right? So then that's why you have sometimes blast reduction, but you don't have full normal blood count recovery. So the response of about 90% CR and durability of about 4 to 6 months is what is, what you would expect to see for patients with, who come on our trial. And of course, we saw 4 out of 7, and in our go forward dose, 2 out of 3 MRD negative CR. MRD negativity is not consistently being published for AML patients. So for relapsed refractory AML patients, sorry. So that's something I would expect is even lower when you look at that. Typically you have these many percentage are CR and a subset of them are MRD negative. In our case, all our CR patients are MRD negative.
Got it. And these distinctions, the CR, the CRH, how are those viewed by regulators in terms of the endpoints there?
Yeah. So, regulators obviously go by what's clinically meaningful, right? Both CR and CRH are clinically meaningful. In fact, you could argue, and there have been papers who've shown that outcome data, that any amount of blast reduction and any amount of count recovery can be meaningful for AML patients, right? Especially when consolidated with transplant. But there is regulatory precedence for drugs being approved with a composite CR rate of CR/CRH. And sometimes platelet transfusion independence, which captures another type of incomplete recovery, is also included in that. So there is regulatory precedence for that.
And so, as you said, you showed a pretty high rate of CRs and responses in the patients. What are you learning from the patients that didn't respond?
Terrific question. So one of the things we do is, and we presented that data at AACR, we looked at the different patient factors, and we will so we are looking at 49 different markers by site off. We're also looking at FLT3, CD33, and endomucin levels on those patients. The sample size is too small really to make any sort of strong statement. So we didn't see any trends to the responders being any subtype of AML or any particular type of mutation or genotype popping out. Really, again, sample size is small. But we will continue to monitor. That's one of the things we are doing. Whatever we did see, some trends we saw wherein all of our responders also had FLT3 expression above a certain level. Not all of our responders, I should say 3 out of our 4 CRs. So things like that we'll continue to monitor to see what that tells us as we continue to progress this with further development.
Got it. And you talked a little bit about what's important to regulators. Do you have a sense of what metrics are most important to clinicians in this space?
Maybe I'll share an anecdote with you. We have, ever since we opened our trial, right, from before we even put our first patient on, so much excitement. Every single slot we have, because remember in a phase 1 trial, you often have, you've got to dose a patient, wait, look at their safety and so on. So it's a staggered enrollment, so to speak. We've always run waiting lists because there's just so much excitement from the physicians, first initially for the mechanistic approach. And then as the data started coming in, obviously, the clinical validation of that MOA. After our presentation at AACR, not a day goes by where we are, where either me or someone on my clinical team, someone reaches out. They want to either join our trial, or they want to learn more about their program. So I would say it's been received very well. We've had just terrific excitement for folks to come on our trial, because CD33 is validated. This is kind of a novel mechanism. Our early clinical data is looking good. So quite a good amount of excitement from folks in that community.
And on the safety front, what kind of feedback has there been? How does the safety profile compare with, other therapies in this space for this type of patient population?
Very clean. There's hardly anything to talk about with safety. There's been some heme events, which if you look at our count recovery, when you give the patients the chemotherapy, the counts drop. As their AML response, the counts come back up, right? So some limited heme events, very consistent with what you would expect in this patient population who get flu-RSV.
Got it. And then lastly, on the regulatory front, what are seen as approvable endpoints for this type of patient population?
That's a well-trodden regulatory path. Quite a few drugs have been approved. It's a combination of response rate and durability of response. The most recent ones that went down that path, the menin inhibitors, just again to kind of benchmark, about 25, 30% CR for CR/CRh, 20% for CR. Slightly more when you bring in CRh as well. Durability of CR about 4-6 months. So that was the most recent, right? Menin inhibitors as a benchmark for what got approved and what came before us.
Is there a sense of whether, some of these mutation specific or targeted therapies might have different, trajectories or benchmarks? For example, again, the menin is for a specific mutation type. The broader population that you're addressing with your study, could have a different disease biology there.
Not necessarily in terms of what is an unmet. I mean, there is a huge unmet need, right? Because if you look at the patients, the vast majority of patients who don't have one of these, what are they getting? It's recycled chemotherapy. So the unmet need and the bar remains the same across the board. And what I will say is we target CD33 and FLT3 on the extracellular surface of the cell. CD33 is nearly universally found on the surface of AML cells. So in that sense, we do require all of our patients who in order to come on our trial, they all need to have CD33 positive AML. We don't have a cutoff or anything like that. When we looked retrospectively in the AACR, it did not seem that any minimum level of CD33 was required to correlate with response.
Got it. That makes sense. It makes sense. Maybe so you had all this data that you presented at ASCO, got feedback at AACR, got feedback there. We've now gone through ASCO. There's been a bunch more additional data across the oncology space. Any takeaways or additional thoughts after that in terms of your positioning or treatments in the AML space more generally?
Further confirmatory of how we're thinking of the unmet need in this space and how we are planning our development. There was some additional menin inhibitor data, more mature data came in line with what I've been saying so far. So no surprises for us. And it continues to kind of underscore that we are on the right track in terms of how we are thinking of what would be needed to move our product forward all the way to the finish line.
Got it. Makes sense. And then you talked about translational data earlier. Obviously, as you said, the point of the design is to spare the normal hematopoietic stem cells while hitting the leukemia cells pretty hard up front. So to what extent are the translational data telling you that you're meeting that goal with where you currently are at dosing?
Yeah, great question. So we had a pretty comprehensive correlative poster as well at AACR, but we put some of the key highlights which talked exactly to address your question in our main clinical data. So we looked at two things in our translational poster, right, which goes to the heart of our mechanism of action. One was our LSCs also being targeted, and I just touched on that. We showed that not only are LSCs in G0 phase at baseline, all of our responders had more than a 10-fold decrease in LSCs post-treatment compared to pre-treatment. The flip side of that was our HSCs being protected, HSPCs, right? The hematopoietic stem and progenitor cells. And indeed, that's what we saw. And so in bone marrow, we saw that in all our responders, the HSPCs are either maintained or increased. And we also saw that a normal kind of immune cell repertoire comes out from those cells. We saw that both in the periphery as well as in the bone marrow. And in the correlative poster, what we did is we also zoomed in on endomucin positive HSPCs, and lo and behold, those are again maintained or increased. So, very small sample size, we acknowledge that, but terrific beginning, not only on the clinical side, but on the correlative side, kind of showing that clinical MOA, and that translating to the clinical data in the patients in terms of normal count recovery. So all in all, very consistent. All data is kind of tracking in the same direction.
That makes sense. On the endomucin, I think one of the patients also had endomucin expression in the AML. Can you comment on what that means and what that's telling you going forward?
Yeah, so maybe, before I talk about the clinical data, if I may step back for a second in terms of how did we identify endomucin as the ideal target. So what we did in our early discovery is with our bioinformatics group, we put together a multitude of publicly available databases and so on, transcriptomics data, more than 800 AML patients, and endomucin was found in about 1% or so of bulk blasts and about 3.5% or so of the LSCs, right? So we know that it occurs rarely in AML. We did see one patient in our trial who had endomucin in the data presented at AACR. And we are quite focused on trying to see, are we going to see more of that? What does that mean? Is that going to impact our response or not? Because what we're measuring on the trial, of course, is proteomics. We're looking at the protein expression. So, we've got to see, is the protein somehow different from what we detected with the mRNA? Is it, and most important to patients, what is the clinical impact of having something like that? At this point, it's with an N of 1, it's difficult to say, but we will continue to monitor that. And the one final piece I will tell you is, NK cells, when they kill a cancer cell, it's because of the balance of activating signal and receptors are higher than the inhibitory, the signals they're getting. So an endomucin found on the surface of a cancer cell is coming in a background of other activating signals telling the NK cell to kill it, right? As opposed to an endomucin found on the background of a healthy cell. So it's a complex interplay. So it's not just the CAR-mediated killing, but also the inherent NK anti-AML killing. So we are going to continue to monitor to see, first of all, are we seeing endomucin expression or not? And two, how is that correlating with clinical activity, which is the most important measure?
Makes sense. And so next steps, you've shown the data, you've landed on a preliminary phase 2 dose. What is next for the program?
So next for the program, the first thing is to confirm that recommended phase 2 dose, and it's a 3+3, so put 3 more patients, look at the safety, confirm that, and then it's expansion. We have in our trial, we have AML expansion cohorts, which is our focus for this year. We can also down the road go into MDS and other tumor types as well. In parallel, what we're also doing is planning for pivotal by working on our CMC, because our clinical trial is a seamless phase 1 to pivotal, so we can keep adding patients on to define a pivotal data set, so to speak. The key is when will we have pivotal GMP material ready. So in parallel with us moving forward with this trial, we've kicked off, from the beginning of this year, because one thing I do want to highlight is last December we presented 2 out of 3 responses in our first dose cohort, so we knew this program can move to pivotal pretty quickly. So we've kicked off all of the pivotal readiness, which is ongoing very nicely in CMC.
Got it. And how does donor screening come into this?
Terrific question. So one of the things which we believe differentiates us is we've built in donor screening from the beginning. So initially, our donor screening criteria was, of course, very much focused on what are the donor attributes which helps with like manufacturability and so on, as we were transitioning from like preclinical mouse to human scale, right? But now that we've got our data, one of the things that we are looking at, and we will continue to look at, is what donor attributes, if any, and we'll do that for donor, for product, for patient, is correlating with responses. And how can we continue to focus our donor selection strategy so that we can enrich for such attributes if we find them. So that is very much a part of what we do here at Senti. So it's kind of built into our life cycle, right?
That makes sense. And then just in terms of the patient selection, we talked about, a few different markers here that might have different expression across patients. To what extent could incorporation of, let's say, a threshold level of FLT3, CD33, or endomucin become relevant here?
At this, based on the data we presented at AACR, we did notice a trend that 3 out of our 4 responders had FLT3 levels at 5% or higher, right? So it certainly was something we're keeping an eye on. At this time, we don't believe that the signal is sufficient for us to modify or require any additional eligibility criteria defined around these things. But we'll continue to monitor what we see in our data and the expression levels in a retrospective fashion. And if the data tells us that we really need to be going in that direction, because remember, we did see one of our responders had low detectable FLT3. And we have shown preclinically as well with our bivalent CAR, either CD33 expressing AML or FLT3 or both, the CAR activity is equipotent, so to speak. So we'll continue to monitor, and then if the data tells us we need to do something different for eligibility, of course, we'll be open to that.
Got it. And then in terms of data sharing, what kind of data set would you next be looking to share with us?
The way we've approached our data sharing is we've always focused on releasing meaningful data sets, right? So back in December 24, our first dose cohort, 2 out of 3 patients responded, and we felt that was meaningful, that you started a trial, your first dose cohort, you're seeing responses. Then in AACR, we had identified our preliminary RP2D, so we are almost at the tail end of our dose finding. We felt that was meaningful. And we also put in our correlative and preclinical because we were seeing concordance with our MO, those data sets for our mechanism of action. So in terms of what's next, we've not publicly said that, I don't believe, unless Jay or Tim can correct me. But this year we are saying, we are targeting about 20 patients or so overall on trial, where we expect to, like, one good endpoint from a trial perspective would be, when you confirm your RP2D or you have some data from an expansion cohort or something like that.
Got it. And then maybe moving beyond the trial itself, obviously right now we're in a pretty challenging time for the biotech markets. Can you guys talk about how you're managing your cash burn and the strategy there?
Sure. So in a word, diligently, I guess. We're very cognizant of the prevailing capital markets and the headwinds and challenges therein. We did report that we had about $34 million in net cash on the balance sheet at the end of our fiscal and calendar first quarter for this year. What we've said is that cash balance will be sufficient to fund operations in the early part of next year. So we obviously pay very close attention to our expenditures and everything we're doing respective of our clinical and preclinical pipeline, and doing everything we can to extend our cash runway as far out as we possibly can.
And then I guess just beyond the Senti 202 program, obviously the technology can be leveraged in other types of assets and modalities. Can you talk a little bit how you're leveraging that either internally or with partners, and what the functionality there is? Mm-hmm.
Yeah, as Jay mentioned, we are focused on Senti 202 and moving that forward. As also echoing what Connie said, we've seen a lot of clinical interest. So our definitely our near-term focus in terms of what we're spending dollars on and executing on is generating that next data set, and we think that's going to be meaningful to the market once that data is out there. But as I alluded to in the beginning, these logic gates do work in T cells. We think, in solid tumors, there's a lot of need for, better, multi-antigen targeting. And so we've previously shown, and actually even on our website, there's some information there around being able to use this sort of logic gating strategy to protect against solid tumor target toxicity. As an example, we have an example in there using CEA as the cancer target, which is a really great colon cancer target as an example, but also is known to be expressed on some healthy epithelial cells. And we've been able to protect against those epithelial cells in a very similar way as in AML. So we do anticipate being able to build programs like that. We have some in our pipeline that we haven't fully disclosed the details of, but they are there, and we think that's a great opportunity for Senti to potentially push those forward, or to work together with partners who could find these very interesting co-development opportunities.
Fantastic. So maybe in the last couple of minutes or so, I wanted to ask if there's anything else that investors might be missing about Senti.
I think probably a big thing from our perspective is this is not a conventional cell therapy product that's going after the same targets or indications that many others are pursuing. We have really taken effort here to go after indications that essentially conventional modalities, cell therapies or antibody-based therapies aren't able to do. And so I think we believe we're quite unique in that sense. We are isolated from sort of the mass competition that's going on around some of these indications. And we think, with data that's coming out and will be coming out as this trial completes, we have the opportunity to showcase that further, that these logic gates can make a huge difference. And then as we continue to build up that data set and success in the clinic, we certainly look forward to that logic gating technology, making it sort of more broadly applicable to other areas.
Understood. All right. I think that brings us to the close of our session. So thank you all again for participating.
Thank you.
Yeah.
Great. Thank you, Geulah.